Everybody doing okay? We ready to get through this? Today's going to be a simple day. All I need is your attention for about 45 minutes. I'm going to run through the PowerPoint that we didn't get through last time. That's our fungal diversity. And like was up last time, there's a lecture quiz about fungus. That's lecture quiz number 4. We ready for the last fungal PowerPoint? Let me fuel up so I'm ready. We left off our discussion of fungus last time on this slide, and we had just really introduced the 5 clades of fungus. This one comes with a nice picture of each of those different types of fungi. We'll see more examples as we jump into this. We'll start at the top with the most primitive fungal group, those Chytrid fungus. And one of the themes that we will see as we go through each of these 5 fungal clades is that they are each going to get their name, like these funny names like Chytridiomycota or Zygomycota, Glomeromycota, Ascomycota, or Basidiomycota. These names are all named after their sexual spore-producing structures. I got to qualify that for a second because we're going to see there's 2 ways in which these fungi can make spores. That's how they get around. They produce those tiny little spores that are blown by the wind to a new location. There's 2 ways that they can make spores, either asexually through mitosis. That's easy to do, doesn't require a partner. Or the sexual spores are going to be produced by meiosis, and that does require a partner. The structures that they produce when they make those sexual spores are what give these particular groups their name. We'll see this as we get into the slides. We'll start with the Chytrid fungus at the top. The name of this clade is Chytridiomycota or Chytridiomycetes. I said mycota and mycetes. Those are 2 endings that you commonly see at the end of fungal clades. So these Chytrid fungus are the most primitive type of fungus. They're the most primitive of those 5 clades. They typically are just unicellular, or they may have just a few of these cells that will string together and form very simple mycelium. You can see one here. You can occasionally find this Chytrid fungus in pond water. We sampled some pond water last time. It is associated with pond water because this is one of the fungus. I can say that one more time. It is associated with pond water because this is the only of the 5 fungal clades to still be considered aquatic. The rest of these 4, remember, have made some adaptations to life on land. And we even see this when we look at pictures of these under magnification. Their spores still retain a flagella, which indicates they had to navigate an aquatic environment, not just blown by the wind. I think that's the last slide on the Chytrid fungus. So let me add another detail to that. Maybe one other thing that's noteworthy about the Chytrid fungus is that you hear them in the news when talking about the fact that we are currently living through a time in which the diversity of amphibians worldwide is on a steep decline. We're losing amphibian diversity all over the place, and one of the main reasons, in addition to habitat loss, are these Chytrid fungus. Amphibians, as we will learn, they have moist skin, and they actually do some gas exchange across the surface of their skin. They can breathe through their skin, which makes it a nice, ideal environment for fungus. They like a moist environment. So the amphibian skins provide that. And we will also learn amphibians, they need a puddle of water to reproduce. They don't have internal fertilization, so females just find a puddle of water and drop their eggs, and then males will drop their sperm in that puddle of water. The sperm has flagella on it that have to swim over to the egg. So amphibians need a puddle of water to reproduce, just like this particular group of fungus needs a puddle of water to reproduce, which is likely why it comes into contact with so many amphibians and is one of the reasons why those amphibians are in such a steep decline. Chytrid fungus. Now moving to the next group of fungus, the Zygomycota or Zygomycites. We call these the bread mold fungus. These look something like this. This is the genus Rhizopus, We have some slides of it in lab today, and you'll see these hyphae that have little stalks, and at the end of the stalk, there's a little cap that's full of spores. There are two ways, like I said, that spores are produced, either sexually or asexually. Those particular spores are produced asexually. We'll see that on the next slide. But one note about the Zygomycota, their hyphae are that particular type of hyphae we introduced last time that have no septa between the nuclei. So there is no separation from individual nuclei. That cytoplasm and nuclei can kind of move freely throughout. Here's another picture of this bread mold. This rhizopus, as we will see, has just about completely taken over this particular loaf of bread. So how the fungus is going to move to a new slice of bread is by putting up these tiny little spores. These are mostly asexually produced spores, and we can zoom in on that structure to see what's going on. Here that structure is. I said it looks like maybe like a lollipop on the end of a little stick here. That stick is the hyphae, and this round lollipop looking structure is what we call a sporangium. It's full of spores. So that name kind of is helpful. Sporangia is full of spores. Those are a bunch of the sporangia that we see, just like we saw in the previous picture. Sporangia are spores that are produced by mitosis, by asexual reproduction. This is an easy way of producing spores, but it's not where the group over there gets their name of Zygomycota. Instead, this is the structure that produces spores through meiosis, through sexual reproduction, and this requires a partner. So you can see two separate hyphal threads here have come together, and together they've produced this one cell that has diploid nuclei in it. This is the spore-producing structure called a Zygosporangium. It's where the group gets their name, Zygosporangia, Zygomycota. Here's another picture of that, just in a series of pictures. The first picture shows right after the fusion of cytoplasm. So we've got two complementary hyphal threads that have fused their cytoplasm. We call that plasmogamy. It led to this heterokaryotic cell. That's a cell that has multiple sets of haploid nuclei that have not yet fused. The fusion of those haploid nuclei, we said that process was karyogamy, and it resulted in diploid zygotes. Here is the cell that is now housing those diploid zygotes. Zygote, we said, was the first diploid cell that forms. Now, in all of us, we were at one point a zygote, a single diploid cell, and then we quickly divided into multiple diploid cells. Fungus don't do that. Fungus don't have a multicellular diploid stage. I'll say that one more time. In the life cycle of fungus, we don't ever see a multicellular diploid phase. When we get into plants, I know I'm jumping the gun to talk about plants, we'll see that some of the first primitive plants don't have a multicellular diploid phase. Plants have to spend some time adapting to life on land and slowly develop this multicellular diploid phase. Fungus don't have it at all. Fungus, as soon as this diploid cell forms, it immediately goes through meiosis and we have haploid spores. So spores are produced two ways, like I said, either through asexual reproduction. This doesn't require any separate mycelium, just this one mycelium can do it all by itself. It just needs enough nutrients, and it can use that nutrients that it stores up to produce these sporangia. Again, spores are going to carry it to a new slice of bread, hopefully. It lands in some place where there's plenty of nutrients, and if it does, it can continue to germinate and grow into a new mycelium. It's quite happy going through asexual reproduction. But if it runs into another hyphal thread that is of a separate mycelium, so the color difference here is showing you that these are two hyphal threads of different mycelium that have just landed on the same loaf of bread. And maybe in a romantic story, this red one here catches the eye of the blue hyphal thread. And it's inevitable at this point. They're going to run into one another. And so the first thing that happens in this little courting ritual of the two hyphal threads, maybe the music is playing right here, Plasmogamy occurs, and that fusion of cytoplasm happens without the fusion of those nuclei. So it creates this cell that we said was heterokaryotic. It's got multiple haploid nuclei from different hyphal threads. We're going to see that some fungi stay in this heterokaryotic stage for longer. In Zygomycota, it's just a single cell that is heterokaryotic. Then those haploid nuclei fuse through that process of karyogamy. Now we have diploid nuclei. And like I mentioned, there's no multicellular diploid phase. So as soon as these diploid cells form, they immediately go through meiosis and produce haploid spores. So two ways of producing spores, one through meiosis, one through mitosis. Again, the sexual spore-producing structure is the Zygosporangium. It's where the group gets their name, and that's what we're trying to find in lab today. I want to see a Zygosporangium and then a Sporangium. Sexual spore-producing structure, asexual spore-producing structure. We okay with that one? All right, moving on to the next one. Glomeromycota or Glomeromycetes. We refer to these as the root fungus. I talked about these things last time as an association between a fungus and the root of a plant. This term root fungus let me say that again. This term mycorrhiza, that micro just means fungus and rhiza means root. So it's an association between a fungus and a root. The prefix in this case, endomycorrhiza, tells us that this relationship between a fungus and a root actually penetrates to the interior part of the cell. Endomycorrhiza, we can see these hyphal threads that have actually penetrated the cell wall of the roots and are now inside that root cell. It's like that Alien movie where there's something inside the cell, but in this case, it's not all bad because what these hyphal threads are actually conveying to that plant cell are additional water and additional minerals. This plant is hooked up to a sponge basically underground. Can convey extra water and minerals to it if it's tapped into those hyphal threads. But it's not all good either because those hyphal threads are in turn going to extract some of the sugar from the plant. We'll see the plant has to pay back about 25% of the sugars that it makes to its fungal partners down there in the ground. Endomycorrhiza is when the fungi have penetrated the inside of the cell. Ectomycorrhiza refers to this fungus association with the root that's outside the root, on the surface of the root. Ectomycorrhiza. You can see all these surface hyphal threads that are kind of tangled around the root of this particular sapling. Down here you can see saplings that have an association with a root fungus have a much quicker start to their growth than saplings that lack this association with the fungus. And you can imagine if you're one of these plants and a tree falls in the forest and now temporarily there's a little bit of sunlight, the canopy has opened up. It's a race to see which plant can grow the fastest to claim that spot in the canopy and then produce the most sugars. So plants that have this association with the fungus are going to grow more rapidly and claim those open spots in the canopy. So that's great. Now that plant's going to be able to produce a bunch of sugars, but again, it's going to be paying back some of those sugars to the fungus. We'll see this in the video. So I will skip to the next group here, the Ascomycota or Ascomycetes. We refer to these as the cup fungus. And it's really kind of on the fence in terms of which of the last two groups of fungus are the most common. I will say that the cup fungus are the most varied. There's a wide variety of Ascomycota. And another thing that's noteworthy about these Ascomycota is that when we go to look at lichens, we introduced lichens last time. We said lichens are part fungus and part algae, but I may not have said that it's not a perfect, it's not an equal balance when we're talking about lichens. A lichen is 75 to 80% fungus. It's dominated by the fungal part and only 20 to 25% photosynthetic partner. I have to say photosynthetic partner because most of the time that photosynthetic partner is a green algae, but sometimes it can be cyanobacteria. It doesn't always have to be a green algae. But it's always part fungus and then part photosynthetic organism, and the fungal part is the most dominant part, like I said, about 80% of the organism. And all of the fungus, like we said, that make these lichens are part of this Ascomycota group, the cup fungus group. And when you look closely at the lichens, even under that dissecting scope, you'll see a bunch of these cup structures. Again, think cup fungus. I'll just say Basidiomycites. These are also considered to be an extremely common group because these are the ones that you would encounter if you go to the grocery store, like I did over there and bought a bunch of different types of mushrooms. They're all Basidiocarps. These are examples of Ascomycota, the sac fungus. We said it was the most diverse group, so a lot of these produce these easy to recognize cups. Inside those cups, if we look the slides today, there's a slide of a Basidia. It's a cup fungus, and when you look at it under magnification, you see inside that cup there are the spore producing structures, those dark staining cells or the spores. We'll see them in a second. Also included in this group of Ascomycota are the morels. Those are little depressions. Those are the cups in which you would find the spore producing structures. And then this down here is yeast. It's also part of the sac fungus. Again, an extremely diverse group. We also saw a video last time when we introduced these fungal types. We saw that one of those fungal types is a predatory fungus that actually can hunt living organisms like this nematode worm by weaving these little lassos out of hyphae, secreting a chemical enzyme that mimics the smell of food, and it tricks these nematode worms into thinking they're going to get a meal. So they wiggle their way through those lassos looking for where the food's at, and then it actually clenches tight and kills the nematode worm. Extracts its nitrogen from the dead carcass of the nematode worm. Some characteristics about Ascomycota. They have, again, when we think about the hyphal structure, these have a separate type of hyphae than we saw in Zygomycota. These hyphae are separated by those septal walls, but they don't have any caps in the pores. So the nuclei are sectioned off, but the cytoplasm is not sectioned off. It can move through those pores. Our examples in lab are this Basidia slide and the Saccharomyces slide, the yeast slide and the cup fungus slide. This is what the yeast slide is going to look like. There's not going to be a lot of detail on it, but you can see that little depression of that little cup there is where the spore producing structure is going to come out of. Here is, when you look at the Basidia slide, you're going to see this cup structure. You're going to need to look at it on the 4x objective lens, as zoomed out as you can possibly get. This structure that produces the spores is called an ascocarp. We will see this association with, like, this ascocarp produces these ascospores, and the name of the group is Ascomycota. So there's some redundancy there in the name. Ascomycota, ascocarp, ascospores. If we look closely at that ascocarp, the fruiting body's name is an ascocarp. So let's look at the spore producing structure. That little, it looks like a columnar-shaped cell. That's going to be called an ascus. It has inside of it 8 ascospores. So there's an ascus. We can see 8 ascospores. Here's another picture of an ascus. There's another, the tip of an ascus is also poking into this slide. Each ascus, each columnar-shaped cell is going to house 8 of those ascospores. Some redundancy there. A lot of Ascomycota, a lot of ascocarps, a lot of inside this ascus, that one cell inside the ascocarp. We're going to see this in this slide, but just let me jump back to this one large picture of it. This large ascus is going to be the only cell in which karyogamy happens. Remember, karyogamy is the fusion of 2 haploid nuclei to form a diploid zygote. That diploid zygote, like we said, immediately goes through meiosis and yields 4 haploid cells. We've got 8 ascospores here, so what happens is those 4 haploid cells go through one round of mitosis, 4 haploid cells become 8 haploid cells, and that's how we get these 8 ascospores. Before I get any further, there are, like we were talking about in Zygomycota, there are ways that you can produce spores sexually and ways that you can produce spores asexually. We skipped over Glomeromycota. We'll see that in the video. But now we're down here in Ascomycota. I'd mentioned that the name of the clade comes from the sexual spore-producing structure, the Ascocarp. But there is an asexual spore-producing structure called a conidia. They're not named after their asexual spore-producing structures, so that term conidia doesn't match with the term Ascomycota. Okay, so now to look at this entire life cycle of this Ascocarp. We'll start with the asexually produced spores. So once again, 2 ways of making spores. We've got spores here and we've got spores up here. These asexually produced spores came from that structure that we called a conidia. And those conidia came directly from a single hyphae that had stored up enough nutrients, enough to produce these spores that could carry it to a new location. And as long as they land in sufficient nutrients, that spore can grow to a new hyphae. Again, each of those nuclei are still going to be haploid nuclei. In order to get to a diploid cell, 2 of those complementary hyphae are going to have to catch eyes. Again, they're not using male or female, but they're using different colors, red and blue. So these 2 haploid hyphae from different mycelia strayed too close. Maybe they caught each other's eye on the train, whatever. We've got another one of the music starts to play. And these 2 hyphal threads share their cytoplasm. Notice, I wish there was a few more pictures right here, so just let me describe a little bit about what's going on. We kind of pick up the picture right here where we can see these hyphae that are dikaryotic. They've got 2 haploid nuclei in them. And that's what I wish there was a little bit more of the picture here. I wish they were showing the entire mycelium, that entire web of hyphae. That entire web of hyphae would be made out of these dikaryotic cells. And only a few of those dikaryotic cells, the ones that are in the ascus, those are the only cells in which karyogamy is going to happen, where those 2 haploid nuclei actually fuse to form a diploid zygote. We said the diploid zygote is very short-lived. It immediately goes through meiosis and produces 4 of those haploid spores. We said each of those haploid spores goes through 1 stage of mitosis. So we go from 4 haploid cells to 8 haploid cells. And it's those 8 ascospores that are housed in those columnar-shaped ascus that basically pack that structure called an ascocarp. So out of the top of an ascocarp come ascospores, out of an ascus. A lot of redundancy there. But again, these groups are named after their sexual spore-producing structures. In this case, it's an ascocarp. That was a mouthful. Here's one of those morels. Again, there's that little cup shape. Truffles belong in this group. It's, like I said, an easy to recognize group whenever you can see these cup shapes. And we should easily recognize some of those cup shapes when we look at the lichens in the lab activity. This, in fact, is one of those fruticose lichens, and you can see those cup shapes that are all throughout this particular lichen. Screaming Ascomycites. Okay, introduced Ascomycites. That leaves the final group down here, Basidiomycites, or Basidiomycota. These have, once again, a different type of hyphae. I had to use this same picture of hyphae, so let me use my words up here to distinguish this. Basidiomycota, they have hyphae that are separated in terms of their nuclei, so they have septate hyphae that have caps on their pores, so that nuclei does not flow freely from one cell to the next. Capped pores on these septate hyphae. Another thing, like I just mentioned in the previous slide, Ascomycota and Basidiomycota, they both have these really large, long-lived Dikaryotic Mycelium. So if we can see that web of hyphae underneath the ground, and we could look inside each cell of that hyphae, there would be 2 haploid nuclei that just don't fuse. The group is named after their sexual spore-producing structure. So right there, the beginning part of this clade, Basidiomycota, that term Basidia is going to be used a lot in the next few slides. In fact, this, what we would call a mushroom, is technically called a Basidiocarp. Instead of Ascocarp for Ascomycota, it's Basidiocarp for Basidiomycota. So I was talking, I even have each of these things labeled as a mushroom, but technically they're each Basidiocarps. This is an oyster mushroom. But if we look closely at that Basidiocarp, you can see those gills on the underside. If we could pluck out just one of those individual gills and put it underneath the microscope and look closely, what we might see is this little, that's a single cell that's kind of a club-shaped cell. That club-shaped cell is called a Basidia. It's where this group gets its name, club fungus. That's supposed to look like a club, this Basidia. It's a special cell because only in that one cell do those haploid nuclei fuse into a diploid zygote. As the story has been, as soon as that diploid zygote forms, it immediately goes through meiosis and produces these 4 haploid spores. Those are called Basidio spores. If I can zoom into that same picture, there is that club-shaped cell called a Basidia. It's only in that one cell, only in that Basidia, that 2 haploid nuclei fuse. That process that we called karyogamy, it leads to the diploid zygote, which immediately goes through meiosis and forms those 4 Basidio spores that we can see right there on the image. Even right up there, there's another one of those Basidia, and you can see Basidio spores dangling from it. So, sexual spore-producing structure, the Basidia, that's where the group gets its name. There are asexual spore-producing structures on these Basidiocarps. They are also called conidia. They are rare, and they're extremely varied. So we don't look at specific examples of conidia in the Basidiomycota. They're named after the Basidiocarps because that's the sexual spore-producing structure. Here's the graph that shows what's going on here. We don't see any asexual spore-producing structure on here. But what we do find are 2 of these haploid hyphae. So these are individual mycelia. They're, again, using different colors to indicate that these are separate mycelia. But in this Basidiomycota, the haploid hyphae are relatively short-lived. As quick as they can, they find a complementary hyphal thread and they fuse their cytoplasm. So plasmogamy is common, and we end up with these Dikaryotic Mycelium. This is what I was hoping was on that previous life cycle slide. These particular Basidiomycota, they have really large Dikaryotic myceliums, like the size of multiple football fields put together. Sometimes they're described as potentially one of the largest living organisms because they're again, they can be acres in diameter. And you never see the actual body of the fungus because it lives underground. You only ever see its reproductive structures that it pushes up through the surface when it's trying to move to a new area. So the mycelium, as well as these reproductive structures, they're going to be made out of these dikaryotic cells. Each has 2 haploid nuclei in it. And only in those special cells called Basidia do we ever expect those 2 haploid nuclei to fuse. Again, haploid nuclei fuse into a diploid nuclei is karyogamy, and those diploid nuclei immediately go through meiosis and produce these 4 haploid spores. Same thing, what I like about this picture is that it shows that underground mycelium is really made of dikaryotic cells, one cell that has 2 nuclei in it, 2 haploid nuclei. So the mycelium is made out of dikaryotic cells. The entire fruiting body is made out of dikaryotic cells. All of those cells have 2 haploid nuclei in them. And it's only those Basidia, the last ones on the tips of those gills, that those 2 haploid nuclei ever fuse to form this diploid cell. That diploid cell immediately goes through meiosis, and we've got 4 haploid spores. Those haploid hyphae are short-lived. They quickly fuse through plasmogamy to form these large dikaryotic mycelia. So I liked that one because it showed a little bit more of that large mycelium underground. Examples of Basidiocarps would include these earth star fungus. I think we might have plugged these at one point last time, and I'm about to show a video with fungus in it, and I'm immediately reminded that the video I'm about to show today does not include the video of the earth star, so I may have to bring some additional videos next week, but that's okay. If you look over here, it's kind of dark in the picture, but these are the outside of these earth stars. They have a really thick exterior coat that peels away in the fall. This usually coincides with the rainy season. These, now that they've opened up, that thick exterior has peeled away, and what it has revealed is an extremely thin and delicate membrane with a little pore on the top of it. This little ball is full of spores, and if a rain droplet happens to hit the surface of this membrane, as just happened to this one, it disturbs the surface of that membrane, and that vibrates the air inside here and sends spores shooting out the top. It looks like a puff of smoke, but it's really just thousands of individual spores that are now going to be carried by the wind to a new location. So puffballs and earth stars are examples of Basidiomycota, and those are spores rather than smoke. We'll see shelf fungus as another example. In fact, the limbs that I brought in last night, one of them has some shelf fungus on it. And that's not a bad thing. You typically see these shelf fungus on older trees. There used to be this parking lot that I would go to. Maybe you've seen one of these things as well. Maybe think about your apartment complexes or things like that, where sometimes they'll see like a tree branch, like a limb has fallen off of a tree and it's exposed some dead wood. Let me take a step back further than that. I know we haven't gotten into plants yet, but let me use my arms here to try and represent the trunk of a tree, like a big thick trunk of a tree that's about the diameter of my arms here. What's kind of fascinating about the trunk of a tree that's even this thick around is that the only living layer of cells in a tree trunk this big are the layers of cells that are just underneath the surface of the bark. Again, I know we don't have a tree in here yet, but just maybe close your eyes and imagine peeling through the bark of a pine tree. Once you get through that bark, there's going to be a layer of cells, a single layer of cells about the width of a single sheet of paper. That single layer of cells just underneath the bark is the only living layer of cells in that plant. It's called the Vascular Cambium. We're going to see it in Unit 4. Once you go through the Vascular Cambium maybe into this center part of the trunk of the tree, that solid wood there is all dead wood. The fungus likes to eat dead wood. It doesn't eat the living tissue at all. So the whole point I'm trying to say here is it always breaks my heart whenever somebody puts like tar or paint on the fungus. You typically see the shelf fungus or bracket fungus eating on some of the dead tissue of the tree. And I think maybe people don't know that it's just eating the dead tissue and not the living tissue. They think maybe it's killing the tree, and so they try and kill the fungus, and that's terrible. The tree actually benefits a lot from that fungus. There are, in Cameron, I have these bee traps out where I'm trying to trap some bees. And one of the bee box locations is on a golf course, and I was out there the other day, and there's an oak tree right next to this bee box that got blown over by a windstorm just about a month ago. And that happens to these oak trees as they grow kind of tall. They become top heavy, and if the ground becomes too soft and the wind's too strong, they can blow over. Older oak trees we will see that have this relationship with the shelf fungus. The shelf fungus will remove that core of dead wood, and it leaves behind this hollow tree trunk that's much lighter and less top heavy, less likely to topple over in a windstorm. And we'll see it also does some other interesting things, like provides homes for forest dwelling animals. That's also beneficial for the tree because they drop their residue, their droppings, which sounds nasty, but the plant loves it. It's additional fertilizer for that plant. Anyway, this is a shelf fungus. It's just eating the dead tissue of the plant. This is another picture that I took probably 10 years ago on, this is how good cell phones used to take pictures. This is a shelf fungus that's eating on a pine tree. Again, it's only eating the dead tissue. This next picture, again, is not very clear, but you can kind of see daylight coming through the top of this tree. This is just an opening in the tree, and that shelf fungus has gone all the way through and hollowed out this tree. It's now a home for an owl. Okay, last slide, but we're not quite done. There is some moving parts that I wanted to show. And I guess before I go to the video real quick, there's just a quick reminder of the different types of lichens. We're going to see 3 different types of lichens. They are 75% fungus, 25% photosynthetic organism. The fungus dominates the partnership there, so it's the fungus that determines the shape of the organism. You can see that with those cups that pop up telling us Ascomycota. There again is another one of those cups that's even called an Ascocarp. You can see Ascospores that are forming. So this is, like we said, 75 to 80% fungus. Those fungal threads, those act as a sponge to pull moisture out of the environment and convey that moisture to the green algal cells. Those all by themselves would be aquatic. Those algal cells need to stay in water or they would dry out. The only reason they've gotten out of the water and survived is because they found a home sandwiched in between these layers of hyphal threads. So what the algae get out of this relationship is a home that prevents them from drying out. And in turn, what the fungus gets out of this relationship is a sugar source. This is the slide of the same thing. When we look at some of these slides, the prepared slides, they have hit them with a stain. So the red staining dots here, those are actually the green algae that have taken up this red stain. Here are the 3 types of lichens. You'll see all 3 types under the dissecting scope back there. The crustose lichen grows like a crustwood on the surface of the bark. It even grows on granite rocks, or you see it growing on tombstones as well. There is the foliose lichen, which is broad and leaf-like. And then there is the bushy one. This is kind of a fuzzy picture there, but this is the fruticose lichen, the bushy one. Okay, so we've introduced our lichen types. Let me grab something from the back just to show this real quick. I brought this big branch in here that's got a bunch of different types of lichen on it. And you can see that, just kind of as an example of what he was just talking about, if I grab, that's fine too. In thinking about these lichens, this is obviously a much larger branch, so it's been around longer. So the lichens on it have been growing for a longer amount of time than the lichens that would have been growing on this smaller branch, which has been exposed to the environment for a less amount of time. You can see much larger lichens on this larger branch. If you look at this larger branch also, you can see there are some of this shelf fungus that is clearly put out some of these reproductive structures. This is not the body of the fungus. The body of the fungus is inside here dissolving the plant tissue. If this was, a tree that didn't have an association with the fungus, it likely would be a really hard branch, but now that the hyphal threads have been in here to kind of dissolving this plant tissue, I know I'm going to make a mess here, but you can actually just get in here and shred the plant. It's external digestion, like we said. So I'll clean all this stuff up. If you actually take some of this dissolved plant and put it under the dissecting scope back there, you can see some of the hyphal threads that are in here, kind of dissolving some of that wood. Or you can also see the lichens that are here. These are large enough that just by looking at it with the naked eye, you can see those little cup shapes, which are kind of telling you that the fungal part here is one of those Ascomycota. So there's a lot of stuff on this. I'll close this video off and stop that buzzing noise. And I'll say that will conclude the lecture part of this class. So the next thing that's on the list is our fungal diversity lab activity. There's 3 steps to this.